Microorganisms are organisms typically too small to be seen with the human eye. Most exist in the micrometer or nanometer range. While the human eye can resolve objects down to about 100 micrometers, most microbes are significantly smaller. Bacteria typically measure around 1 micrometer, while viruses are much smaller, measured in nanometers. These life forms represent immense biological diversity, classified into cellular groups like bacteria and fungi, and as cellular entities such as viruses and infectious proteins known as prions. In microbiology, the standard metric system allows us to measure life at an incredibly small scale. We start at the millimeter, but most microorganisms exist far below this threshold. Most bacteria are measured in micrometers, typically around 1 micrometer in size. This scale is fundamental for viewing cellular structures. Viruses are significantly smaller, often measured in nanometers. A typical virus might be 100 nanometers wide, requiring electron microscopy to be seen. Understanding these units of measurement is crucial for microbiology, as it defines the tools and techniques we use to observe these tiny life forms. All cellular life is divided into two fundamental types based on their internal structure: prokaryotes and eukaryotes. Prokaryotes, which include bacteria and archaea, are the simplest cellular forms. They lack a membrane-bound nucleus. Eukaryotes are significantly more complex. They possess a distinct membrane-bound nucleus that houses their genetic material. This group includes protists, fungi, plants, and animals. The primary distinction lies in internal organization. While prokaryotes maintain a streamlined, compact structure, eukaryotic cells utilize organelles to compartmentalize functions. Bacteria are ubiquitous, single-celled prokaryotic organisms. Unlike eukaryotes, they lack a true nucleus, keeping their genetic material in a concentrated nucleoid region. A hallmark of most bacteria is the cell wall composed of peptidoglycan. This structural matrix provides protection and maintains the cell's shape in various environments. Bacteria occupy nearly every habitat on Earth. Their metabolic diversity allows them to thrive in extreme conditions, from deep-sea vents to the human microbiome. While many bacteria are beneficial to life, aiding in digestion and nutrient cycling, others act as pathogens, causing disease in hosts. Bacteria are categorized primarily by their distinct cellular shapes, a concept known as morphology. This structure is key for identification. The first common shape is the coccus. These bacteria are spherical, appearing as individual circles or grouped together. Next, we have the bacillus form. These bacteria are rod-shaped, which provides a larger surface area relative to their volume. Finally, spirillum and spirochete bacteria are curved or helical. This unique morphology often assists in motility within liquid environments. Recognizing these structural differences is a fundamental step in clinical microbiology for identifying specific bacterial species. Bacterial metabolism is defined by incredible flexibility. Bacteria have evolved diverse pathways to obtain energy and carbon, enabling them to inhabit almost every environment on Earth. Some bacteria are photosynthetic, such as oxygenic cyanobacteria. They harness sunlight to fix carbon dioxide into chemical energy, mimicking the fundamental process found in plants. Chemoheterotrophs obtain energy and carbon by consuming organic compounds. They break down environmental nutrients through respiration or fermentation. Lithotrophs derive energy from inorganic environmental compounds like hydrogen sulfide or ammonia. This unique capability allows them to survive in extreme environments where organic matter is scarce. By diversifying these metabolic strategies, bacteria maintain the chemical balance of our planet's biosphere. Archaea are unicellular prokaryotes that, while superficially similar to bacteria, represent an entirely separate domain of life. The primary difference lies in their cell wall composition. Archaea utilize pseudopeptidoglycan, which is structurally distinct from the peptidoglycan found in bacterial cell walls. Archaea are famous as extremophiles, capable of thriving in environments with extreme temperatures, pH levels, or salinity that would be lethal to most other organisms. Notably, despite their abundance, current research indicates that no species of archaea are known to act as human pathogens. Protists are a diverse group of eukaryotic organisms that do not fit into the categories of plants, animals, or fungi. The primary defining trait of all protists is the presence of a membrane-bound nucleus within their cells, which houses their genetic material. Protists include algae, which are generally plant-like and photosynthetic, and protozoa, which are animal-like and often motile. Protists exhibit incredible structural diversity, utilizing specialized mechanisms for movement like cilia, flagella, and pseudopods to navigate their environments. Algae are a diverse group of photosynthetic eukaryotic organisms. Unlike bacteria, they contain a nucleus and specialized organelles. A defining anatomical feature of algae is their rigid cell wall, composed primarily of cellulose, which provides structural integrity. Algae perform a critical role in global ecosystems by generating oxygen and serving as primary producers that anchor the aquatic food web. Beyond nature, algae are economically significant. They provide agar for laboratory research and are widely used as thickening agents in food production. Protozoa are a diverse group of single-celled eukaryotic organisms. Unlike plants, they are heterotrophic, meaning they must consume organic material to survive. They are defined by their unique methods of movement. Cilia are short hair-like projections, flagella act like long whips, and pseudopods are temporary cytoplasmic extensions. In aquatic ecosystems, protozoa play a vital role in nutrient cycling, feeding on bacteria and organic debris. However, some species have evolved to become parasitic pathogens in humans and animals. While some protozoa are essential components of our food webs, others cause significant diseases. They remain a complex and critical field of study within microbiology. Fungi are eukaryotes, meaning their cells possess a defined nucleus. A defining structural characteristic of fungi is their cell wall, which is composed primarily of chitin. Unlike plants, fungi are non-photosynthetic. They cannot synthesize their own energy from sunlight. Instead, they are heterotrophs, absorbing nutrients from their environment. We categorize fungi primarily into two forms. Yeasts are unicellular, while molds are multicellular, growing in long filament structures. Fungi serve vital roles globally. They are essential ecological decomposers and are indispensable in industrial pharmaceutical production. Yeasts are a group of unicellular fungi that play diverse roles in our world. Unlike molds, which are multicellular, yeasts typically reproduce through a process called budding, where a new cell forms as an outgrowth of the parent. Beneficially, yeasts are essential in food production. Through fermentation, they convert sugars into carbon dioxide and ethanol. This process is the secret behind light, airy bread and the creation of various alcoholic beverages. However, yeasts can also be pathogenic. Candida albicans is a well-known example. It normally resides in the human body as a commensal organism. When the host's immune balance is disrupted, it can grow uncontrollably, leading to infections such as oral thrush or vaginal yeast infections. Molds are multicellular fungi consisting of long, branching filaments known as hyphae. These structures allow them to grow efficiently across organic surfaces. As decomposers, molds play a critical role in the ecosystem. They secrete enzymes that break down complex organic matter, recycling nutrients back into the environment. Beyond their role in decay, molds are highly valued in medicine. The discovery of penicillin from the mold, Penicillium chrysogenum, revolutionized our ability to treat bacterial infections. Helminths are parasitic worms that represent a unique bridge between macrobiology and clinical microbiology. While most are multicellular and visible to the naked eye, they are central to the field of infectious disease. They are studied in microbiology not because of the adult worm, but because of their microscopic life stages. Their eggs and larvae are frequently detected using standard laboratory microscopy. Helminths are broadly classified into platyhelminths or flatworms, which include flukes and tapeworms, and nematoda or roundworms. Both types have distinct life cycles, often requiring specific host environments. In clinical settings, identifying these organisms is critical. Lab technicians analyze fecal or blood samples, looking for these microscopic signs of infection, proving that size does not define clinical relevance. Helminths serve as a vital reminder that clinical microbiology encompasses more than just single-celled life. integrating macroscopic parasites into our understanding of global health. Unlike bacteria or eukaryotes, viruses are acellular. They lack the organelles and metabolic machinery required for life independent of a host. Their structure is remarkably simple, a protective protein capsid surrounding a core of genetic material, either DNA or RNA. Viruses are obligate parasites. They remain inert until they encounter a host, hijacking its cellular machinery to replicate and propagate. By overriding the host's normal functions, viruses can cause diverse diseases, ranging from the common cold to severe viral infections like Ebola. Prions are unique, infectious agents composed entirely of protein. They lack DNA or RNA, which distinguishes them from all other biological pathogens. Normal proteins serve vital cellular functions. However, prions are misfolded versions. This change in shape allows them to convert normal proteins into more infectious prions, leading to a chain reaction. Prions are significantly smaller than viruses, making them incredibly difficult to detect and eradicate. They represent a fundamental challenge in clinical diagnostics. In humans, prion infections lead to severe neurodegenerative conditions like Creutzfeldt-Jakob disease. The accumulation of these misfolded proteins results in rapid and fatal cognitive decline. Microbiology is a vast scientific domain, organized into specialized subfields focused on distinct types of organisms and their interactions. Bacteriology centers on the study of bacteria. Mycology examines fungi, while protozoology investigates various protozoa. Parasitology focuses on helminths, and virology is dedicated to the study of viruses and their replication. Finally, immunology is integrated into these studies to understand how the host immune system interacts with these diverse pathogens. These disciplines collectively provide a comprehensive understanding of microscopic life and global health. The modern clinical laboratory operates in a rapid cycle. Starting from patient sample collection, we process and analyze microbial content using molecular diagnostics. Clinical scientists analyze the battle between host and pathogen. Understanding this interaction informs the choice of targeted antibiotic or antiviral therapy. Ultimately, clinical microbiology is not just about identifying organisms. It is about providing the data that saves patient lives and drives public health strategy. Microbiology research is bound by strict ethical standards. History contains grave violations, including non-consensual human studies that highlight the necessity of constant ethical vigilance. Today, all research must follow institutional review board oversight. These boards review research protocols to ensure subjects are protected and risks are minimized. A cornerstone of modern ethics is informed consent. Participants must fully understand the nature of the study, the potential risks and benefits, and their absolute right to withdraw from research at any time. Through rigorous IRB oversight and the requirement for informed consent, microbiology ensures that scientific progress never comes at the cost of human dignity. Ethical integrity is the foundation of modern scientific discovery. Researchers must navigate strict protocols to protect the rights of human participants and ensure the humane treatment of animals. In human research, informed consent is mandatory. Participants must understand all procedures, risks, and benefits. Protecting their rights, confidentiality, and autonomy is non-negotiable. Animal research is guided by the IAC-SEEC, or Institutional Animal Care and Use Committee. This body reviews all protocols to minimize pain and ensure research animals are treated humanely throughout the study. Scientific breakthroughs are only meaningful when conducted with the highest ethical standards. Maintaining honesty and accountability ensures that human rights and welfare remain the priority in every experiment. Throughout this series, we have categorized the microbial world into distinct groups: prokaryotes lacking a nucleus, eukaryotes with organized cellular structures, and the complex acellular agents like viruses and prions. It is critical to remember that microbiology extends far beyond pathology. While we focus on disease prevention, the vast majority of microbes are fundamental to global ecosystems, sustainable industry, and human health. Looking ahead, the field is evolving through advanced molecular diagnostics, biotechnology for sustainability, and interdisciplinary collaboration. By adhering to rigorous ethical standards, researchers continue to unlock the potential of these microscopic organisms for a better future.